Hello, my name is Terry Powell, and I am the Community Relations Officer here at Lawrence Berkeley National Lab. I'd like to welcome you this evening to Berkeley Lab Friends of Science. For the past 20 years, Dr. Barcellos-Hoff has been a cell biologist doing research in the Life Sciences Division of Lawrence Berkeley National Laboratory, where she currently serves as Deputy Director of the division. Barcellos-Hoff earned her undergraduate degree from the University of Chicago in biopsychology, a field of research in which human behavior is studied as the product of interactions between the brain, the body, and the environment. Her PhD from the University of California at San Francisco was in experimental pathology. She came to Berkeley Lab in 1986 to do postdoctoral research in cell biology under Dr. Mina Bissell, the first scientist to link tissue-specific function and dysfunction in cancer to the, quote, extracellular matrix, end quote, or ECM, a support network of proteins surrounding breast cells. Her field of expertise is the effect of ionizing radiation on tissue. In 2003, NASA awarded Barcellos-Hoff a $10 million grant for a 5-year study of the cancer risks posed to astronauts by radiation in space that our atmosphere normally shields us from. Her interdisciplinary team has developed one of 5 national specialized centers of research in space radiation biology. Experimental findings by her research group have shown that exposure to ionizing radiation creates a microenvironment in the tissue that surrounds breast cells, which can cause even non-irradiated cells and their progeny to become cancerous. However, she has also uncovered aspects of tissue response to radiation that inhibit cancer. This apparent paradox is the topic of her talk today.
This apparent paradox is the topic of her talk today. Thank you. Mary Helen.
Thank you, Terry. And thank you all for braving the rather nasty weather. I must say, we usually show this slide of the view from the top of the hill so that everybody can appreciate how gorgeous it is looking down from Lawrence Berkeley Lab, but today is not one such day. So I wanted to talk a little bit about why we study ionizing radiation and what the context is that we study radiation as a carcinogen. I want to preface that by first announcing, of course, that I'm a PhD, I'm a mouse doctor. I don't study humans, really. I study the fundamental mechanisms leading to carcinogenesis in a variety of different models. And with that information, our hope is that we will be able to better understand human disease, better be able to intervene in it, and perhaps even develop some therapeutics based on our knowledge of the fundamental biology. I'm not going to talk too much today about our NASA work, which is unfortunate because actually NASA really inspires people's interest. The idea of intergalactic cosmic rays and how we're going to understand the consequences of that radiation exposure is really quite interesting. But maybe in a year's time or so we'll have enough information that we can discuss that in more detail. So let's start with just some basic premises about concepts about carcinogenesis, and then kind of evolve those in a more global fashion. So the paradigm that's kind of been the major motivator in the last 20 to 30 years in cancer biology is that there's multiple stages to carcinogenesis. You start with a normal tissue, and what I'm going to depict here is an epithelium. We'll talk about epithelial cells primarily because 90% of cancers arise in the epithelial tissues of the body. Not in every epithelial tissue. It's quite interesting, actually, which tissues actually develop cancer and at what rates. And what I'm going to be talking about is the epithelium of the breast. So my major interest is in breast cancer. So this just depicts a little cross-section of an epithelium in the mammary gland. And the idea with multi-stage carcinogenesis is that there's an event called initiation, and that event occurs in a single cell. Now think about this, because actually it's quite interesting if you just do the math. There are 10 to the 14th cells in the human body. Over the course of your lifespan, you will generate 10 to the 16th cells. 1 in 3 people develop cancer over the course of their lifetime. So actually, while we think of cancer as being a very widespread disease, at the level of the organism, of the human being, 1 in 3 times 10 to the 16th cells is not that frequent event. But do we really understand the events that lead to actually clinical disease? The idea here is that there's a single event that occurs in a single cell, and upon expansion of that single cell, you now have a group of cells, and that is called promotion. So anything that, well, it's thought that things that lead cells to proliferate, create more of themselves, will now expand that population. During that expansion, there's the probability that you will induce additional genetic changes, and that's called progression. So now I've changed the color of the little nucleus up here that has the event that has occurred to initiate the cell. Now there's a cluster of cells here, and over here what we have is a cluster of cells with multiple colors indicating they have a lot of different changes in them, and it's those kind of changes that lead to the ability of those cells to escape from the normal tissue bounds, invade the surrounding tissues or even distant tissues, and therefore cause clinical disease. So with that multi-stage carcinogenesis paradigm, what we're focusing on is the events within a cell. Within a cell, as if that cell had kind of a life of its own. It's kind of like thinking about individual behaviors as being de novo, arising just from thoughts, right? Rather than being a consequence of your upbringing, your genomic, your genetic heritage, and your environment, the context in which you're behaving. So likewise, so that indicates this actually takes years to decades before you actually see cancer in general. When we talk about radiation carcinogenesis, the general idea has been for many, many years is that radiation causes that initial event or some other kind of genetic change. So that because radiation can cause DNA damage, those DNA damages, if misrepaired, can lead to mutations, and that's the basis for the action of radiation as a cancer-causing agent. But what we'd like to review is the idea that these events that occur here are not really occurring in isolation. Cells are incorporated into a community of cells. As I indicated, what we're going to be looking at is the mammary gland. This is a mouse mammary gland. These are the epithelial cells within this little red ruffled colored. These are stromal cells, and it's the interaction between these two cell types that we're going to be talking about primarily. So the idea is it takes a tissue to make a tumor. That a cell doesn't become a tumor. There are no such things as single cell cancers. It really requires these cells within the epithelium to recruit the cooperation of surrounding tissues components, different cell types in the tissue. So what I'm going to talk about is kind of how that might occur. So if we go to how do you get from a single cell to a multicellular organism. There's actually lots of different levels of control, and I'm just diagrammed a couple of cartoons to indicate some of the levels of control that we study as cell biologists. So you start as a single cell, everybody does, but even a tissue. Then that tissue, that cell interacts with something called the extracellular matrix that Terry mentioned in her overview. The extracellular matrix is a network of insoluble proteins that the cells are either embedded in, or in the case of an epithelium, they sit on top of a specialized extracellular matrix called the basement membrane. In addition, there is signaling that occurs between cells. So as you go from one cell to two cells, an epithelium functions because it creates a barrier. The skin, the gut, the mammary gland are each sheets of cells. In order for them to form sheets, they have to interact with each other. And they do so by actually producing proteins that actually link up along their lateral surfaces. Then you have an additional level of control through the microenvironment, which is what I call extracellular signaling. Okay, so these are the signals coming from the outside of the cell, and they're interacting with the proteins on the inside of the cell. Now, these extracellular signals can come from their neighbors, a juxtacrine response. They can come from the stroma, which is a paracrine response, or they can come from distant organs, endocrine responses. So the hormones that you produce in your pituitary affect the mammary gland to influence the production of milk. So you have several different layers of extracellular signaling that impinge upon these individual cells. And then it's together that all of these things lead to the functional differentiation and formation of the functional unit in the given tissue. So in the mammary gland, I'm going to depict it as a little hollow sphere, which is a little bit of a simplification. But if you can think about it, the mammary gland is actually a duct system in which the milk is produced in little alveolar units, which is a hollow sphere at the ends of these large ducts, and milk during pregnancy and lactation is produced by the alveolar cells and then conducted through the ducts to feed the offspring. So I'm just going to take a little snapshot, and we're going to focus on these alveolar units. So if we think now about kind of how do you put these two things together, one way to think about this is one that was suggested many, many years ago, actually. And it's a very simple idea that is actually very easy to kind of grasp. There's two things that you need for cancer. First, you need the seed, which for the most part we consider to be changes in the DNA. And those changes can occur and expand, but they only do so within the appropriate soil. So the tissue is the soil. So you need these changes to occur in a conducive environment. So one of the ideas that my research is based on is that normal tissues suppress cancer. And they actually do so very, very effectively. Otherwise, we wouldn't have that relatively rare incidence of 1 in 3 times 10 to the 16th cells actually going on to form a cancer. And I'm very interested in how this occurs, so I'm going to talk a little bit more about that. Now, the corollary of that is that abnormal tissues can promote cancer. So how does that occur? We're going to talk a little bit about that. And that's what actually leads to carcinogenesis, is the cooperation between the genetically altered cells and their ability to either recruit these abnormal cells, induce the abnormality, or to have some exogenous agent that does both. So my hypothesis for this work is that systems, this is a new word in biology, systems biology is a wonderful thing. It's a brand new field. And what it attempts to do is to take all the reductionist biology that we've been doing, which is cataloging the changes in the genome and the proteins that are being expressed, and our ability now technically to measure those across huge data sets. So 40,000 genes at a time, 30,000 proteins at a time, and now go back to a generalist mode of scientific endeavor, which is to tie everything together. So when we talk about systems biology, we're talking about trying to integrate the information across different levels of organization. And my hypothesis is that the system, i.e. the tissue, the organ, the organism, respond in an orchestrated fashion to limit the damage of cells at the molecular level. So if you go back to that idea that ionizing radiation can cause DNA damage, well, that's true. There's no doubt about it. We've studied it pretty extensively for the last 40 or 50 years, and it's no doubt that that can happen. But in the same breath, we must also recognize that radiation might be changing how or that the tissue can also be sensing that in ways that we need to further study. So the way to think about systems biology, if you were trained earlier than the last 5 years, is that we have physiology, cell biology, and molecular biology that have all been well studied in the last, forever. And what we're now trying to do is link these different levels of organization using this new terminology, systems biology, which really requires a way of thinking about how these are all linked together. So this is an example of how systems biology works. This is actually from a website that says systems thinking. Systems thinkers consider that a system is a dynamic and complex whole, interacting as a structured functional unit in equilibrium, homeostasis essentially. A system is a community situated within an environment. So this could be a community of cells, it could be a community of organisms, it could be the environment itself, i.e. an ecological system. So this is not restricted to biology, it's a way of thinking about science. Information flows between the different elements that compose the system. So information in the context I'm going to be talking about will be these signals that go between cells and between cell types and between organs. What distinguishes a complex system from a merely complicated one is that some behaviors emerge as a result of altered relationship between the elements. Now this is a really interesting idea. It's essentially going back to this idea. So can you describe a person by looking at its genome, his genome, her genome? Can you describe a tissue by looking at the component cells? Can you describe the biology of cancer just by breaking down into its bits? We're very good at breaking down bits. What we have difficulty with is actually putting it back into a functional unit. So what we're trying to do is begin to understand these behaviors that emerge as so that the whole is more than the sum of its parts. So here's an example. A person breathes. A lung exchanges gases, but a lung cell does neither. So breathing is an emergent phenomena, a property of the system rather than of the parts. So it's a pretty good example of how we can't really understand all the biology by just looking at single cells. So what we will want to do is apply that same principle to cancer biology. And I think there's some evidence that cancer may well be an emergent property of a tissue, something that evolves or is expressed as a consequence of minor perturbations rather than global changes. And the example that I like best of this kind of idea is actually from a paper published by Hal Moses just a year or two or so ago. Very interesting paper. We're going to come back to this idea, this particular protein that's indicated up here. But essentially, what they did here is, while I told you that the genomic change occurs in the epithelial cells, what they did in this paper was to engineer a mouse which could no longer respond to a growth factor in these supporting cells, the stroma, the fibroblasts. And what they found was that mouse, even though it had only lost the ability of the fibroblasts to respond to this growth factor, actually developed cancer in both the prostate and the forestomach within 6 weeks of its life. Now that's actually a surprising finding, and hopefully by the end of the lecture you'll understand why that's so surprising. So let's go back to radiation. It always helps for me to kind of give you a little bit of context in regards to dose. Radiation doses are very hard for us to grasp. It's helpful if you have some kind of context to put it into. Keep in mind that humans are exposed to over 5 orders of magnitude of radiation exposure, starting all the way with our environmental exposure, which in the case of average North American is about 350 millirem a year. So that's just part of life, right? What we're thinking about here is cancer radiotherapy. So radiation is used to treat cancer. And when you do so as a physician, you use doses anywhere from 3, so the unit of dose I'm going to refer to today is gray. Hopefully this will give you some context for understanding what a gray does. So in cancer radiotherapy, you can get 3 gray to ablate your bone marrow if you're going to have a bone marrow transplant, or if you have a brain tumor, the brain might receive a total of 80 gray. That's in fractionated exposure. What radiation biology tends to focus on is about anywhere from 0.1 gray to 10 gray. And that's just, 10 gray is certainly a lot. It's enough to kill 99.99% of the cells in a petri dish for most mammalian cell systems. But that's the area that radiation biology has focused on because it's pretty strong dose. It's easy to see effects. You can get a little bit of a sense of what kind of dose that is, and that this little nubbin there indicates the dose that would, if received by an individual acutely, would result in death of 50% of the individual. So it's about 3.5 gray. Now that's one dose, whole body, over and done with. Okay, that's not likely to happen to anybody. But of course, we do have the survivors from the A-bomb exposures, which received doses between that, about that much, well, actually more. But these are the survivors, and they are somewhere about 3 gray to about 0.1 gray. So, and then there's an area that we're very interested in, which is when radiation is used, is studied to, in terms of its ability to cause cancer. And again, the focus here is on somewhat lower doses. So now we've gone from 100 to 10 to 1 gray. So we're going down in logs of dose here. And remember, I said there are 2 more orders of magnitude before you get to essentially no exposures in humans. So I'm interested in this range of doses because the typical mission on the International Space Station the astronauts will be receiving about 0.1 gray over the course of their time there. And in our NASA studies, we're also interested in the estimated dose range for a 3-year mission to Mars, which will be more on the order of a single gray, but again, protracted over 3 years at a time. So those individuals won't have the same kind of response as somebody who receives an acute dose of radiation. So hopefully that gives you a little context about radiation doses. And what we're going to be talking about today is really the range from, let's see, about down here, 0.1 gray to 2 gray, the doses I'll be talking about today. Actually, no, I take that back. Some of my animal studies, we go up to 4 gray. Now, mice are more resistant to radiation. We can expose them to 4 gray, and they are perfectly happy. Interesting, isn't it? So, the question that we want to pose is how does the breast respond to damage to the DNA? Radiation can cause mutations, i.e. the seeds that initiate cancer. Can it also alter the soil, i.e. the microenvironment, affecting whether cancer progresses? Now, the way we actually think about this is based on some studies that I did when I first started my own laboratory at Lawrence Berkeley Lab, which is I took a mouse, I irradiated it, I took out the mammary tissue, and I looked at various proteins I found interesting. And one led me to another. And essentially this little cartoon shows you the very dynamic nature of the protein changes that you could find in the epithelium of a mouse and some of the stromal cells. So I'm not going to go over those in great detail, but what I want to tell you is that what was remarkable about this study is that we were able to see changes in the microenvironment very rapidly. Essentially, as soon as we took the animal out of the X-irradiator, we could take it and dissect out the tissue and find some events that occurred within an hour of exposure. We also found that the radiation responses were global. It wasn't happening in a mutational manner, which would be very intermittent, but rather every cell was responding. It was extremely sensitive in that some events, again, we could detect at total body doses of 0.1 gray, but it was tissue specific. So we compared the liver to the mammary gland to the skin. In each case, we found that it was rapid, global, and sensitive, but the proteins involved were very different in each case. So each tissue had a slightly different response to the ionizing radiation. It was radiation quality dependent. That alludes to our work with for NASA, where the radiation quality in the in space is quite different. And what I'm going to talk about a little bit later is one particular event that, that we noted that turns out to be quite interesting, both in terms of its contribution to breast cancer, but also in terms of its contribution to radiation damage. So I'm going to come back to this protein called TGF-beta, which we consider to be the center of the universe. Now, I say that somewhat, well, with a fair amount of skepticism, because of course there's no one single protein that's the center of the universe. It's just the center of the universe for my laboratory. So the hypothesis is that the system, i.e. the tissue, organ, or organism, respond to damage caused by radiation at the molecular level. And what I wanted to understand was through this microenvironment changes that we're seeing, that's what I consider the system, is the response mitigate or augment the deleterious cellular effects of ionizing radiation. So in order for us to ask that question, we created a radiation chimeric model. Now, what do I mean by that? The way to test whether or not the effects on the microenvironment are affecting the epithelial cells is to separate the two, irradiate the host, the stroma, the microenvironment, or the fibroblast, and then put in add back to the system the epithelial cells. Now, how can we do that? We can do it by taking advantage of the mammary gland, and I'm going to show you exactly how that happens in the next slide. We can look at bone marrow transplants. That's quite frequently done, actually. It's used therapeutically, where you ablate the bone marrow and then add back unirradiated cells to the host. Or you can do it in culture. You can co-culture irradiated cells with non-irradiated cells, or tissue fragments, or you can even use something called a microbeam to actually irradiate single cells that you can see, and then watch for the observe the effects in both the irradiated and non-irradiated cells. So the idea here is you take the host, you radiate that, you add donor cells back to that host. So that makes it the chimera. And in the case of the mammary gland, I'm going to show you that in just a second. In this case, we're doing the bone marrow transplant that goes back into the tail vein. The tail's disappeared there. Or in the case of a co-culture, you can take cells, irradiate those, and then add non-irradiated cells to the tops of them. Those all allow us to now study the various contribution of radiation effects in different compartments. So here's a mouse mammary gland, highly stylized. Essentially, a mouse mammary gland is a very flat, little thin disc of tissue. It's mostly fat. And in particular, it's mostly fat prepubertal. So in a young mouse, which in the case of mouse aging is between birth and 3 weeks of age, the epithelium, which I am afraid has disappeared here, is white. This is, just ignore this, this is just a landmark, okay? So this is going to be the lymph node, and it actually just marks that it's about a quarter of the way from one end to the other end. So here's where the nipple is, and the epithelium grows out from the nipple like that. Okay, so under the hormones of puberty, you develop this beautiful ductal tree, which is not so beautiful in my cartoon here. But the idea is that that ductal tree, since it begins at puberty, if you just come in to a 3-week-old mouse and cut out that piece of tissue, now what you have is just the stroma, the fat pad. What's remarkable about the mouse mammary gland, which was actually described by Ken Diome here at UC Berkeley in the 1950s, is that at any point in the animal's life, if you now inject epithelial cells back into that little flat disc of fat, they will grow and form a new ductal tree. So that allows us to make mix and match experiments between, epithelium of one sort and stroma of another. And what Ken Diome did was actually take these cells and expose them to the carcinogen, and then, like chemical carcinogens, and other people did it with radiation, and then show that those cells gave rise to tumors. And what I did was reverse the experiment. So this is the way we reverse it. This is our group of mice in which we've removed the epithelium at 3 weeks of age. It's a very simple little surgery. Close them back up and they're happy as clams. Then we take what is a mammary epithelial cell line. Okay, so these are cells that normally grow in culture, but they have a remarkable outgrowth potential, so that when we put them back into 3-week-old mice, they form those ductal trees. We now think these were originated at Baylor University, or Baylor College of Medicine in Texas by Dan Medina. And at the time, it was just a spontaneous event. We now think that this cell line maintains stem cell properties, and that's why it can recapitulate the normal mammary gland outgrowth. So interestingly, though, although they make a normal outgrowth in a 3-week-old animal, they have a mutation in a very potent oncogene called P53. Now, we don't have to get into exactly what P53 does, but in 1994 it was named the molecule of the year as being the guardian of the genome. And so what's quite frequent in human tumors, in human cancers, is that P53 has mutations in it. So it no longer performs its guardian function. And what we found in these cells, or what we thought we'd do with these cells, is that since they had this mutation in it, maybe they were actually capable of making cancers, but they were suppressed from the normal tissue environment. And that's why when we put them back into the normal mice, they just make outgrowths. So now we took them and we did an experiment where we transplanted them into normal mice and to irradiated mice. And now these mice were those that I mentioned had a significant dose, 4 gray of radiation. So that's not a tickle dose by any means. mice are perfectly happy. We've kept them out for 2 years of the full lifespan of the mice. Their hair turns a bit gray, but then so does everyone's. What we do then is transplant these epithelial cells into the host that have either been irradiated or not irradiated, and then we score for what happens. And what we found, obviously, was tumors. So this is actually a real fat pad stained with a dye so that you can see the epithelium. You can see it's branched in various and complex ways. And this is what happens when we put it into an irradiated host. We get a lot of tumors. So the cells are still capable of making the outgrowths, but they also make tumors at the same time. And we quantified that. We actually showed that they were from those cells that we transplanted because they had this aberration in p53 that was easy to detect. So now we quantified that, and we compared the control mice to the irradiated mice for 2 features. One is the percent of fat pads with tumors, and as you can see, 75% of the animals that had been previously irradiated now had tumors, whereas in the control host, only 15% of the animals developed tumors. So actually that was kind of interesting because we thought none of these cells could make tumors, but it turns out that if you inject the cells into a 3-week-old mouse, they never make tumors. If you inject them into a 10-week-old mouse, which is the condition under which we did these experiments, then they make tumors, which when it again says that what's important about the capability of these cells to make tumors is the environment. So whether it's 3 weeks or an adult, or whether the mouse is irradiated, makes a tremendous difference in the ability of the cells to form a tumor. We also measured the size of the tumor, and so this is the tumor volume, and you can see that in the few mice that formed tumors, the tumors were very tiny. They were just like the little size of a lentil, whereas in the irradiated mouse, we got much larger tumors. That means they rapidly progressed. So those data were very interesting, and this just shows here that it didn't make any difference if we irradiated the mice 1 day before we transplanted or 14 days before we transplanted. The effect of radiation on the host, on the microenvironment, was quite persistent. Now I just said host or the microenvironment, and you might be wondering, well, radiation has a global effect. Which can we actually attribute this to an effect locally on the microenvironment, cell-cell interactions, or is it a global systemic effect affecting perhaps the endocrine signaling from the ovaries or the pituitaries, or it could be an immune response? All of those are possible, but what we did in this experiment is to take our mice and shield them so only half of the mammary glands, well, left versus right, were irradiated, right? And then we injected into both sides, and what we found was the tumors only formed on the irradiated side. So since the other side shared the same systemic milieu, we think that this is a local effect on the microenvironment. So from these experiments, we concluded that carcinogen-induced microenvironments, i.e. radiation-induced microenvironments, the radiation can induce cell phenotypes, behaviors of cells, that can lead to an altered microenvironment, and that these cells that are involved in this are actually, in this case, in the stroma or the host. These carcinogen-specific microenvironments affect the frequency and the features, like the size of the tumor, of malignant progression, and that this actually can constitute a third class of carcinogenic action, distinct from those influencing genomic change or proliferation. Now, what this makes a very interesting target, if you may, to think about in terms of preventing cancer. See, the radiation effects on the epithelial cells are going to be random. You're going to generate mutations of all sorts, and how do you know which mutation is going to occur in this particular individual? In contrast, we think these carcinogen-induced environments are really part of this physiological response, really a systems response. It's kind of like wound healing. In this case, it's wounding without any blood. But if it's like wound healing, it's organized. And if it's like wound healing, it's orchestrated. And if it's like that, then we probably can figure out ways to interfere with it so that we can suppress the abnormal environment and control the potential for cancer cells. So, that is one conclusion from this, that these can promote the neoplastic progression of the initiated cells. But of course, these are mouse cells. And one of the questions we have, well, is this only a function of mouse behavior? Mice are quite different than humans, although they share a lot of similarities, at least in terms of mammary gland biology. So we had to also ask this question, does radiation affect on the microenvironment alter human cells? So to do this, we took a page from my postdoctoral work with Mina Bissell several decades ago at this point. Kind of scary. But what we did was, in my postdoctoral work, we took mice and we took out their little mammary glands. We diced them up into little bits and isolated those bits into cells that we could then culture in Petri dishes. Now, if we do this, actually, it's not very satisfying. The cells sit there for a bit, they proliferate a tiny bit, and then they just kind of poop out. You certainly can't get them to make milk. On the other hand, if we take these same cells and do something quite different, which is give them an extracellular matrix, give them that basement membrane that I was talking about, then they do something very remarkable. The same cells put onto this extracellular matrix, which is commercially available as Matrigel. Wish I had bought stock in that 20 years ago. What happens is the cells now aggregate, and when they aggregate, they can undergo morphogenesis, in other words, reorganization, until they form that nice little hollow sphere that I was talking about. And indeed, when we tested these for their ability to make milk proteins in response to hormones that are present during pregnancy, these little hollow spheres just would fill up with milk proteins. So it means that they not only were organized in a functional fashion, but they were behaving in a functional fashion. And when we compared, actually, it's probably hard for you to see on this screen, but this is a cross-section of one of these taken with transmission EM, and this is a light micrograph of the mouse mammary gland in cross-section, and they bear remarkable resemblance. Essentially, those are at the same magnification. So we were really able to recapitulate a functional mammary gland in miniature in the Petri dish. So we took a little bit of a page from this study and actually further developed it in Mina's laboratory. She published a very interesting paper which showed that if you take normal cells, normal human mammary epithelial cells now, not mouse cells, and put them into this Matrigel, they form these nice little hollow spheres, which are what this little cartoon is supposed to indicate, the red dots being the nuclei, and the little hollow center right there in the middle, right? But in contrast, if you take tumor cells or cancer cells, put them under the same conditions, they fail to undergo this morphogenetic event. They form aggregates, clusters, and continue to grow. These cells will actually stop growing and maintain a perfectly normal number of cells for one of these acini. So what she concluded from these studies is that the interaction with this basement membrane serves to rapidly distinguish growth and differentiation, which is appropriate in the normal cells, from that proliferation and disorganization of malignant human breast epithelial cells. So what we asked is, well, what happens if we irradiate normal cells? Can they undergo normal morphogenesis? So the question actually goes to our cell-cell interaction. So I remember I said there was microenvironment was important, and cell-cell interactions are important for normal tissue behavior. So we now tested this idea. We took non-malignant human mammary epithelial cells. So these are cell lines, so that's why I don't call them normal. Nothing that's a cell line, which means that it's immortal in culture, is really normal. But they are non-malignant by all traditional assays. And what we did was we irradiated them when they were single cells, and we asked, could they actually form these nice little acini? So this is now images of those acini directly from our studies, and what we're doing here is comparing this top row to this bottom row. And I'm going to come back to this TGF-beta in just a moment, but what we're looking really at is the ability of radiation to change this cell's response to TGF-beta. So what we're looking at across the, let's see, each of these are proteins, the integrins, that are necessary for cells to attach to the extracellular matrix. and they come in different flavors: alpha-6, alpha-3, beta-1 integrin. And as you can see, when you compare the unirradiated cultures to the irradiated cultures, the distribution of these proteins changes. So in the case of alpha-6 and alpha-3, you get a significant reduction in the staining for those. So we're now staining these with specific antibodies, which are just agents to allow us to localize specific proteins. In the case of beta-1 integrin, what happened was that it actually becomes more cytoplasmic. So you see this graininess to the cells. Then we looked at another protein, which I mentioned earlier, the one that serves to link cells together so they can form an epithelium. E-cadherin is what it's called, and it actually does stick out from from the cell surface and link up between cells to make this kind of tight bridge. And it's usually localized between the borders of cells. And what you see in the radiated sample is that it's almost completely missing. Furthermore, if you compare these, top panel, you see that hollow sphere is formed in each one. There's a little tiny hollowness there. And in each case down here, instead of getting a nice sphere, we're getting kind of more of an irregular cluster. We actually measured that and can prove that quantitatively that these were disrupted morphogenesis. They weren't responding to the signals in the same fashion as the unirradiated cells. So we quantified that in a variety of different means, and this just shows you that the radiation by itself wasn't able to have this effect on its own. It needed the cooperation from this extracellular protein called TGF-beta. And it's the two of them together that led to this loss of cell organization and immunostaining for E-cadherin. So we were interested whether this would not only disrupt morphogenesis, but change the behavior of the cells. Because that's what's important. In cancer, you know, there's a couple of studies that were published in Lancet that showed that, you know, if you look at autopsy material from, well, autopsy samples from people who have died in car accidents, if you look at the thyroid, there's something like 50% of individuals have nascent thyroid cancer, yet the incidence of thyroid cancer in that age group is 1 in 4,000. If you look at prostate, essentially compare Japanese men who never develop clinical prostate to American men who do so at a very high frequency of 1 in 5 over the course of lifetime, they have the same incidence of early lesions. And what differs is the ability of the cells to progress in the American individual. So there's some very interesting things about whether these cells are just going to sit there or whether they're going to really behave abnormally. So what we did, and I don't know if you can see this, maybe if we can turn down the lights just for a moment more, you can see that what we've done here is something as a cell biology trick called a scratch assay, and we're asking whether the cells behave normally, which is as an epithelium, which means they should move as a sheet when we disrupt the sheet behavior. Okay? So these cells are growing in a petri dish, and let's see, I think actually that will help a little bit. What we're looking at here at the top is the sham treated cells, and this is a course of time. So what we do is we run a little rubber pipette tip down the center of the monolayer, and so you're looking at the borders over here, and those are sheets of epithelial cells. Then we're looking at, as a function of time, 20 hours and 40 hours post creating that wound. And what happens in the unirradiated cells is that they move glacially, very, very slowly, as a sheet. You see it's a smooth edge all the way along front. If you irradiate the cells, they actually move very little. If you treat them with this protein TGF-beta, they move a lot more, but still as a sheet. But if you treat the cells with both the radiation and the TGF-beta, they will move as single cells, crawling out into that space. And that's exactly the kind of behavior you don't want your epithelial cells to be doing. So that's a feature of malignant progression. So this increased motility of the irradiated cells when they're exposed to TGF-beta would seem to be a bad sign. So what we are concluding from these studies in the human cells is that radiation can also induce a heritable phenotype of behavioral change in human cells. What's really interesting about this is it's not the irradiated cells themselves, but the way we set up that assay for our single cells to grow to these little balls of cells means that the daughters of the irradiated cells are actually undergoing the aberrant morphogenesis. So that indicates that this is actually a heritable disruption of cell adhesion proteins, cell extracellular matrix interactions, and the organization of this little acinus. So this phenotype is really reminiscent of malignant progression and would not bode well if it occurred at high frequency in an intact organism. So what we've concluded from this is that both mouse cells in the intact organism and radiated human cells can undergo aberrant behavior. So, how do we get from that to this? Well, we get from that idea, which I hopefully have convinced you of, to the idea that radiation can actually inhibit cancer by focusing in on one of the components of the irradiated environment. And I'm going to quickly take you through some fascinating biology about the center of the universe, TGF-beta. So I mentioned this TGF-beta as being very prominent in our irradiated mouse studies. And actually we discovered that in 1991. We published our first paper on it in 1993. So we've been working on it 15 years, well, not quite 15 years. TGF-beta was only discovered 25 years ago. And it has turned out to be the most fascinating protein, at least, for many people. There's 25,000 papers on it, okay? Not an easy thing to keep up with. Nonetheless, we have discovered, by using ionizing radiation, some very interesting aspects of TGF-beta biology that were really previously unsuspected. So it's known that TGF-beta is a very effective tumor suppressor. So a tumor suppressor acts to inhibit growth, can act to maintain a quiescent stroma. It can act on the immune system in a way that suppresses carcinogenesis overall. And we know this because we've created mouse models and cell culture models in which TGF-beta is deleted, and it's not a good thing. What our studies with ionizing radiation has revealed a new aspect of TGF-beta biology, which is that it actually is intrinsically linked to DNA repair in epithelial cells. Now that actually is the topic of a whole nother talk. And so what I'm going to show you is just a little bit of TGF-beta biology, and I'm going to summarize just a few pieces of the data that we're just now getting ready to publish. So this is a kind of a cute cartoon. It kind of takes you through all the basic biology of TGF-beta. There's a TGF-beta gene. TGF-beta1 is the founding member of this group of proteins. It encodes for a 360 amino acid polypeptide, which is cleaved intracellularly into 2 polypeptides. This just gives you, don't try to focus on any of the details, it just gives you an idea of how elaborate nature is. Those polypeptides then dimerize, which means that they form cross-links between them. They homodimerize, so you end up now with 2 little pieces. And then those 2 pieces undergo another protein conformational change called non-covalent association, and now we've switched to my favorite little hand model of TGF-beta, where TGF-beta is the active bit, is this small bit here, and it's grasped by latency-associated peptide. And what latency-associated peptide, or LAP, does is confer latency. In other words, TGF-beta is not active, and it's this latent complex that is actually secreted outside the cell. It's actually abundant in the extracellular space. There's TGF-beta everywhere. All cells, as far as we know, can make TGF-beta. It's packaged in your platelets. It's circulating in your plasma. All cells, as far as we know, have receptors, so they can receive the signals from TGF-beta. But the fact that it's secreted in this latent form requires that there be an additional processing step before TGF-beta can send its signal. And that processing step is called activation. Upon activation, TGF-beta then binds to its receptors and induces all sorts of fascinating things. And we're able to actually follow these events in vivo by using antibodies that recognize either the latent complex, LAP, or TGF-beta only when it's been released from activation. So in this next set of slides, we show TGF-beta in this in red, and in green is LAP. And for the moment, let's just ignore RSMAD down here. And what you see is that essentially there's lots of green and very little red until you've irradiated the tissue, and then you see this pretty orange color come up, which indicates the activation of TGF-beta. RSMADs are just a protein that is downstream of TGF-beta, so when you go from a TGF-beta inactive to an active state, the RSMADs increase very quickly. Now that is what happens in mouse cells. In human cells, we can do the same kind of experiment. We now have taken the human mammary epithelial cells, irradiated them 6 days before we took this shot. And what we're looking for here is, again, these SMAD proteins, which indicate the activity of TGF-beta. And as you can see in the irradiated cells, there's significantly more RSMAD activity. And furthermore, if we add in, we can show that this is TGF-beta activity by doing all sorts of neat tricks. So clearly radiation is inducing TGF-beta. What is it doing? So we've decided that TGF-beta is very interesting because now one of the things that we discovered, well, we haven't decided that, but so one of the things we discovered about TGF-beta is this latent complex is not only just sequestering the TGF-beta, so they cannot act with its receptors, but it's actually a sensor. And what it's acting as a sensor for is free radicals. So what are free radicals? It's the thing that everybody tells you in your, nutrition ads and your makeup creams that you want to get rid of free radicals, right? Well, you can't get rid of free radicals. You're an oxygen-breathing person. You're making free radicals all day long. That's the bottom line. But of course, you can keep them down by eating your fruits and vegetables. And it turns out that latent TGF-beta is actually a sensor of those free radicals, or what we call oxidative stress. So as a reactive oxygen sensor, it's wonderful because it's outside the cell, it's there, it's very rapid, you can activate it very efficiently and independent of any kind of other protein complex. And TGF-beta then acts as a signal for this oxidative stress. So when you get a bolus of oxidative stress, which happens during inflammation, during ischemia perfusion, during ionizing radiation exposure, or if you don't eat your fruits and vegetables, then TGF-beta gets turned on, and that causes cells to apoptose, inhibit their proliferation, differentiate, and cause more reactive oxygen. So that makes it a very elegant feedback loop where you can actually keep going until you resolve the normal tissue toxicity of oxidative stress. So its action as a TGF-beta tumor suppressor, this was the really nifty stuff, is that global radiation response is what we discovered is that if you took cells that cannot make TGF-beta and now irradiate them, they fail to undergo apoptosis or cell cycle arrest, which is what they should do. They fail to modify proteins that are signals of DNA repair. And they lacked functional enzymatic activity, which is necessary for the whole cascade to be kicked off. So this failure of TGF-beta signaling in a TGF-beta compromised model that we've created indicates that TGF-beta is actually at the top of the pyramid in terms of how cells respond to DNA damage. And if you don't have TGF-beta, or you can't respond to TGF-beta, then that would compromise your genomic integrity, your stability of the genome, because you wouldn't be able to repair it. And we actually think that's very pertinent to carcinogenesis because almost 50% of all cancers cannot respond to TGF-beta. They've evolved mechanisms to escape from TGF-beta just because TGF-beta does so many important things to keep them from being cancers. So when we look at this, we wanted to test whether this was actually the fact that radiation induces TGF-beta was actually protecting the tissue. So the way we tested that is we looked at these little proteins or little organelles in the cell, which are called centrosomes. Now, you remember from your high school biology that your DNA is packaged into chromosomes, and those chromosomes are only evident during mitosis. And during mitosis, they're divided in half to each of the daughter cells. So when they divide in half, what actually pulls them in half are these little organelles that are centrosomes. They're kind of like the tractor-trailers of chromosome biology. But what's really interesting about centrosomes is that they're deregulated during carcinogenesis. So you can get centrosome amplification, so instead of getting a split normally, you can get a three-way split, or you can get chromosomes that don't follow through to the daughter cells. And so you get multiple copies of one chromosome in a cell. And that's a hallmark of cancer. So you really don't have to follow all the little red arrows down here. All you have to know is that centrosome amplification can lead to tumor progression. And this is what centrosomes look like in a Petri dish. They're these little tiny green spots. And each cell, which is indicated by a blue spot, which is the nucleus where all the DNA is, should have one, or in some cases they should have two. That's because the centrosomes replicate with cell cycles. So this cell is in G1 and that cell is in G2, which means it's about to divide. This cell in the middle is in big trouble because when it attempts to divide, and it has a big nucleus, it has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 centrosomes. This is not a good sign. Okay, so, but we can measure this, and so that's our index of genomic stability. And what we found was when we irradiated our normal mammary epithelial cells, then we could induce these centrosome abnormalities. Okay, that again does not bode well for radiation. But what was interesting is when we added TGF-beta to these cells, that's the red line here, we significantly suppressed the ability of radiation to cause those centrosomes. So why did we add TGF-beta? Because in our Petri dish, there's no stroma to make the TGF-beta. So what we did was we added TGF-betas to support that. So what we're thinking here is that radiation is inducing TGF-beta in part as a control mechanism that then suppresses genomic instability that would be evidenced by these centrosome amplifications. So, to finalize, what I've talked about today is the radiation affects non-target cells, i.e. the stroma. It alters signaling between cells. It can induce a heritable phenotypic change. And that the signaling from this irradiated tissue can either augment the progression of initiated cells or suppress initiation. And it's really important for us to understand under what cases does this proceed versus this aspect predominate. So what we're proposing is that we need to rethink how we do radiation biology. We need to integrate across all these levels of organization so that cellular events are placed in a multicellular or organized context, that systems which are maintained by information in which space and time are factors are incorporated into what we think about radiation as a carcinogen, and that these system responses that constrain aberrant behaviors by signal cells be incorporated into that overall thought process. Because while radiation can give rise to these emergent phenomena, like this perturbation that I showed you in the human cells or in the mouse cells, that results in cancer, by understanding it as a system, we can probably figure out ways to intercede and protect individuals from radiation exposure. So I'm going to skip over that very complicated slide that just says that life is more complicated than we put down on paper, and thank all the members of my group who are shown here in San Francisco last Christmas, I guess. No, last summer. It looks too warm to be Christmas. And my collaborators who are listed over here, and the Department of Energy and NASA who have funded these research. Thank you.
So where do we get some of these TGF-beta? Who manufactures it?
I actually have to make a slide of this, but I have just come across a new face cream that has TGF-beta in it. I actually thought it was really funny because when I came to do a postdoc with Mina, one of the reasons I was interested in working with her is because she had talked about the extracellular matrix. And so when I was finishing up my graduate work, I was struck by this ad in, I think, the San Francisco Magazine, which was for a dermatologist, and the byline was, "I'm having a love affair with collagen." Collagen is extracellular matrix protein, and everybody's talked about collagen in your creams. Yeah, it's a bunch of hogwash. But now we have a TGF-beta containing cream. However, I can't afford it. It's about $600. So, wouldn't that be interesting to find out? Maybe we should start getting the TGF-beta out of our mouse cells and packaging it up to fund the studies. Anyway.
So it seems to me that
I'm going to just step over here because I can see better.
as you go through life,
Mm-hmm.
you really are expected to have cancer. And it's the way your TGF-beta will be able to suppress it so that you can live longer without having that occur. Because the free radicals are always going on as you metabolize everything in your body.
That's right. So I think that's actually a very important premise of how we think about cancer, is that it's not as readily, it's not as frequent an event as we've sometimes think of it. And secondly, that actually, in order for us to be multicellular, we must have figured out, nature has figured out good ways to suppress that aberrant behavior. And the problem, what I was trying to highlight, is that actually, because of the way the paradigm for cancer has been framed, it's sometimes hard to see that, to step far enough back to see those events. And so that's what we're really undergoing right now, is this paradigm shift where we begin to give more weight to looking a little bit above the individual cell level. I think TGF-beta is only one of the molecules. I'm sure there are others. Yes?
You stuck on a slide that you showed earlier, in which you demonstrated with the images that the only way the cells became, cancer-like was with TGF-beta and the ionizing radiation, which kind of is, counterintuitive to what you're saying, that TGF-beta actually is a suppressing mechanism that is involved in this whole system so something cancerous doesn't evolve.
Yes, and I think that's accurate. It's a problem that we're trying to currently figure out which one plays the dominant role. We can demonstrate both occur. You can promote or you can inhibit, and with radiation, and TGF-beta is involved in both sides. And that, unfortunately, is one of the reasons why there are 25,000 papers on TGF-beta, is because it does everything to everyone all the time. The only thing is we don't know. Well, actually, it can do everything to everyone, but we don't know when and where it does it. So this paradoxical behavior of TGF-beta as acting as both a tumor promoter and, a tumor suppressor, but then switching to a tumor promoter role is very important. We think that maybe one of the reasons we can detect it both in these human cell lines is because they're cell lines. And so we're not dealing with completely normal cells. So we're seeing both sides of the coin here. So you're absolutely right. Yes?
In human questions, is there evidence to show that the extracellular matrix is aberrant, depleted,
Yeah. So there's a variety of different observations. Actually, breast cancer is one of those tumors which actually has more stromal cells in many cases than the cancer cells. So it has a reactive stroma. So it's called desmoplasia, where you just have an abundance of stromal reaction in very few cancer cells. So that's one piece of evidence. And then what actually discriminates, what a pathologist uses to discriminate between a carcinoma is actually the ability to degrade the basement membrane and invade into the surrounding tissue. As long as the cells are constrained by that basement membrane, it's called a ductal carcinoma in situ, and it has much lower probability of causing or developing metastasis, for example, than if the cells have already invaded into the basement membrane. Well, so restoration of the basement membrane, that's an interesting question. Actually, Mina Bissell up at the lab is working on a slightly different approach, which is to reestablish the ability of cancer cells to interact with each other. It's kind of hard to make them make a basement membrane. We think that the basement membrane gets degraded by enzymes called proteases, and so you can treat with protease inhibitors. And that actually is a cancer therapy that is quite actively pursued. But what she's proposing is that if you can force the cancer cells to establish communication amongst themselves, you can actually reestablish their normal morphogenesis, and when they form the appropriate kind of structure, they stop growing. So she's shown in that paper that I showed you the snapshot of, that a cell that has lots of genomic changes, as many as you would attribute to any cancer cell, nonetheless, if you take them and force them to reassociate, they will stop growing and so behave normally. So it's very interesting. Actually, the NIH just this month issued a program call to study the tumor microenvironment. And they're going to fund something like $14 million worth of studies just to address that kind of question. So it's a new field, relatively new field. Yes?
Interesting this concept you mentioned about with prostate cancer, pre-form, that you said the Japanese Americans have the same rate of pre-forming, but it seems to develop with Americans, with Japanese.
Right.
Why would that happen? Genetic component,
well, I'm a little more familiar with breast cancer data, but so let me take an example of that. So in Japanese women also, so Japanese men have a low rate of prostate cancer, Japanese women have much lower rate of breast cancer. Yet if those women move to America, their breast cancer rate begins to creep up. So it seems like it's an environmental effect that allows the cells. Environment, I mean by all sorts of environment, not exposure to things, that are harmful, but lifestyle. So cultural behaviors, lifestyle behaviors, and God knows we're still trying to define the others. But so in the prostate, I can't think of well, I don't know the literature well enough to say if they've done a similar study, but I expect that's probably true. So there's combination effects. It's not all genetics. Yes?
In the systems approach, how do you bring back some of the other systems, such as the immune system or inflammation?
Right. Right. So actually, it's a very interesting question. I think there's some fascinating data. Now that people are beginning to turn slightly away from the cancer cell per se as being the only player here, we're beginning to find some really interesting biology that's going on. So over at UCSF, Doug Hanahan and Lisa Cousins have shown that there's a very strong immuno component to cancer. In fact, there's a paper that Lisa just published this last year which showed that in this mouse that's been engineered to have skin tumors by driving a potent oncogene, what actually is required for those cells to become tumors is that the immune cells have to be recruited into the tissue, and those immune cells then process in a way that's pretty undefined at this point, proteins in the microenvironment, and that is actually what allows the tumor cells to grow out. And if you block the immune cells, you completely block the carcinogenesis. So when I say it really takes a tumor to make a tissue to make a tumor, I in my own studies tend to think just in stroma fibroblasts, but there's the vascular system. Judah Folkman has popularized the idea that if you just stop the blood vessels from growing into the tumor, well, you don't have a tumor because they don't get fed. If they don't get fed, they can't grow. Great ideas, and I think we've got to start looking at these other more controllable aspects of the tumor rather than focusing on the mutations. Sorry. Oh, okay.
So how do some of the prescribed drug therapies like tamoxifen affect or influence the cellular activity?
Yeah, tamoxifen is an interesting one. It's thought to be an anti-estrogen, yet interestingly enough, fibroblasts that don't have the estrogen receptor respond to tamoxifen by increasing TGF-beta. So it's quite possible that and there's been a number of studies to try to pinpoint how much that contributes to the effect of tamoxifen in suppressing breast cancer. But probably a number of agents that we prescribe to affect the target cell, the epithelial cell, are actually having actions on other cell types in the vicinity that are required to cooperate. So it's just something that remains to be studied in more detail. Well, I can't see very well.
I know that calorie restriction has also shown to reduce tumors in mice, and I'm wondering if there's any relationship between those results and what you've been doing.
Well, actually, I think that's the topic of Judy Campisi's talk, and she should come and talk to you about aging and the aspects of aging that contribute to carcinogenesis. But she certainly has developed an appreciation of the effects that can occur through the stroma. And there's a growing belief that the association of cancer with aging may be because the suppressive components in the tissue are being degraded or are being lost, becomes less effective. One last question, it looks like. Yes?
I've heard of a study about centromeres. If you will disrupt the centromeres, the disruption in the production of the centromeres will stop the proliferation of the cell division or of the cancer.
So are you thinking centromeres or centrosomes?
Centromeres, you know,
the middles of the chromosomes.
The one that carries out the chromosomes to its opposite end.
Okay, so there's centromeres, which are actually the middles of chromosomes, and there's centrosomes, which are the organelles that move. And absolutely right, if you disrupt the centrosomes by engineering overexpression or deletion of a protein that's important for their integrity, then you rapidly generate aneuploidy and subsequently carcinogenesis. Yeah.
So they are doing studies on that one too?
Yes, actually a new paper in breast cancer just came out on that from Thea Tilstes at UCSF. So I really appreciate your attention and great questions, much better than I usually get. Cool.
Thank you very much for giving Friends of Science a chance to learn more about your research. Let us know when you have your next data published. We'll ask about it.
Okay, we'll
make sure
save.
It's not a rainy day.
We'll do the intergalactic cosmic rays next time. Okay.
Thank you.
Thank you.